We're delighted to have Dr. Venter here, and I'm honored to introduce our president, Ruth Simmons, to introduce Dr. Venter.
But you were going to make some comments first.
There is no time.
There's no time.
Yours, I had some emotional comments. I leave it to you.
Well, emotional comments are perfectly valid, too, so you could come and do that. Okay, well, fine. In deference to all of you who've been waiting for a while for this wonderful occasion, I just want to say good afternoon and thank you so much for coming today. I'm so pleased to be able to join all of you on the first day of this 37th Industrial Partners Program Symposium. And I want to thank Ellie and Soren for inviting me to be here today. I want to extend a warm welcome to members of the Partners Program who are visiting for the symposium. It was not, of course, too many years ago that strong partnerships between the academy and industry were themselves viewed as new paradigms. I want to congratulate our industrial partners and our Department of Computer Science on establishing such a productive and mutually beneficial program. Well, when I was first contacted and asked if I might attend to hear Craig Venter, I was thrilled and very excited, and frankly a bit intimidated to be asked to introduce him. Nevertheless, I'm pleased to do so. Even in a field so crowded with talent, drive, and ambition, his is a record of truly stunning achievement, creativity, and vision. In the late 1990s, as you all know, the International Human Genome Sequencing Consortium was in full swing, working to unravel what had been one of biology's greatest mysteries and challenges. You can imagine the complex organization that such a task, for those of you who are younger, such a task required. Craig Venter stood up and said, in effect, we can do this under one roof, and we can do it in 2 years. His was a bold and visionary stance. With his founding of Celera Genomics in 1998, Craig Venter demonstrated the effectiveness of his approach for assembling the human genome, beginning everywhere and all at once. 300 robotic sequencing machines installed on 3 floors. What must that have looked like? Actually, 300. On 3 floors served up the raw material for what has been called a feast of computation. I love that, a feast of computation. It all worked as Dr. Venter said it would, and as we know, within 2 years, Celera and the International Human Genome Sequencing Consortium shared an extraordinary publishing triumph. 2 simultaneous papers on the sequencing of the human genome, the consortium in Nature and Celera in Science. And most observers believe that the work of each was improved by the work of the other. Of course, with Dr. Venter, there is so much that I could cite from his CV. Decoding the first genome of a free-living organism, that was in 1992. Sequencing the fruit fly, mouse, and rat genomes, 228 publications in scientific journals. I'm sure that number is not correct, right? And counting, I should say. A dozen honorary or more honorary degrees from Taiwan, Sweden, Italy, and the United States. National medals from nations around the world, countless awards, lectureships, prizes, honors, distinguished invited presentations. But one of the things that caught my attention was his current interest in sequencing the ocean. Like a 21st century Charles Darwin on a particularly well-equipped beagle, Craig Venter is following in that great natural scientist's footsteps, cataloging the diversity of life that still teems in the oceans of the world. There is great beauty, I think, in the human drive to understand life, to understand nature, to understand the world. And part of that beauty is the eternal promise, the infinite promise of so much more to study and so much more to come. And that, of course, brings us back to the symposium's discussion of the genome and computational sciences, the next paradigms. Ladies and gentlemen, I say most sincerely that I've been hearing about this man and wanting to meet him for a very, very long time. It is a distinct honor for me and for Brown University to present a singular, outstanding figure among the world's foremost scientists, entrepreneurs, and frankly, visionaries, Dr. J. Craig Venter.
Well, it's clearly one of the nicest introductions I've ever had. I'm now truly glad I came to Brown. I can leave with pride. I'm actually delighted Soren didn't have a chance to give his emotional rendition to be followed by that. Or, you know, I would. I'd rather leave people in tears after my lecture, not before it. So I guess I have to try and. You'd think a bioinformatic symposium would have a better computer set up. But I'm going to tell you about a number of things that are going on at our institute. So this is one of 6 buildings we have in Rockville, Maryland, and another one in La Jolla. So this is the Venter Institute, which has recently combined all the organizations that we formed starting back in 1992 with TIGR. So we have about 535 scientists and staff here covering a wide range of endeavors, and I'll just touch on a few of those and try and put some of this in a context. So this is a review we wrote for the 10th anniversary of Nature Genetics, where everybody was so sure that everything in genomics was just technology development and based on technology. And I've been trying to stress for a long time that philosophy enters this just as much, if not more so, than technology. In fact, I'll argue that what we did with sequencing the human genome was a philosophical triumph, not just a technological one. Simple concepts such as massively parallelism, randomness, were probably more important than any technology, particularly randomness. Massively parallelism is obvious now, particularly those who do computational work, but with sequencing it wasn't so obvious, at least to most. There was a big race to have a single machine that could sequence the human genome. People rejected the early sequencing machines because one couldn't possibly sequence the human genome. But to me it was a very simple concept. If we wanted to double our output, we bought a second machine. And we chased that all the way up to 300 of them. So it became like computing, just the more processors, the more you could do. Randomness was even more important, and this stems back from, even back to my doctoral work at UC San Diego, where I was told this was the early 70s, that it was going to be difficult to come up with something new in biology because it was essentially all known. I mean, it seems so incredibly absurd today, but that was the thinking at the end of the 60s and early 70s. In fact, the US Surgeon General in 1969 announced that we had won the war against microbes, and microbiology departments around the world began to shut down. So science and intelligence seems to go through bottlenecks at times and coming out the other side. But you know, I spent a decade trying to isolate one gene, the adrenaline receptor gene. But just going to a cDNA library and randomly selecting clones, we're able to come up with a very large number of genes in a short period of time. And this is the EST method that we published on in Science in 1991. Just randomly selecting the clones from a library, sequencing them. This paper seems like a small number now because it only had 337 new genes in it. I'm about to show you where in February we're going to publish a paper with 6 million new genes in it, so this is our trivial effort. But back then, getting one gene at a time was a big triumph, let alone hundreds of them. And this advanced very quickly just from random selection to get very large numbers of them. But in fact, it was the random method, the random selection, that led to the first genome. Just breaking the chromosome into tens of thousands of small pieces, randomly selecting those and sequencing them, and then using algorithms to put them back together is what led to Haemophilus influenzae being completed in 1995. Instead of the 10 years it took 1,000 scientists to complete the yeast genome, or the 13 years to do the E. coli genome, this genome took only 4 months to do. Relating to the idea of this conference, we hired a computer scientist, Granger Sutton, who wrote an algorithm to assemble EST sequences. And we thought we could apply it to this other problem, and it was a test. This entire process took only 4 months. And we didn't set out to sequence the first genome, we just set out to try a method. And it ended up being so powerful, applying mathematics to biology, that it ended up taking 10 years off a project. You'll see numbers on these because they put into context some of the later experiments, but it was a big deal at the time to do 25,000 sequences. We had a large facility with a very large number of sequencers. They were not very high throughput machines at that early time, but the computation was not that big of a deal. It's the number of sequences squared, so we had roughly 10 to the 8th pairwise comparisons. But this is an experiment the genome community thought couldn't possibly work at this scale. We wrote a grant and submitted it to NIH in 1994, and it was rejected out of hand because this method couldn't possibly work, and the mathematics wasn't up to it. We followed on, shortly with the first archaea, and so within a year we went from no genomes to all 3 branches of life being covered. And even though we couldn't get funding for the first genome, as soon as we showed this method worked, we had to turn away funding. It came in so fast, and we ran out of principal investigators, in fact, to do organisms. We did most major human pathogens, branched up, in fact, with the next speaker doing the first malaria chromosome, then on to bigger things like plants and insects and human. I think after Haemophilus, the next big breakthrough in this field came with the drosophila genome. And that's where people like Soren played a very key role. Because now instead of 25,000 sequences, we had to do 3 million sequences. So now we had 300 of the biggest sequencers in the world, and we built at the time, in less than a year, the 3rd largest computer in the world, because everybody on our computational team thought that's what it was going to take to do all these pairwise comparisons. So Gene Myers and Soren co-headed the team along with Granger Sutton to build this new set of algorithms for assembling genomes. People were totally certain this would fail. Again, this took only 4 months to do. Less than a year from starting the project, we published the paper in Science with the complete assembly of the Drosophila genome. And after this, everything was obvious. Human was obvious, and now this is the method that every lab around the world uses. Human, it was a scale up a little bit more. We had 27 million sequences. So now we're up to 10 to the 14th pairwise comparisons. And this took quite a while with the initial assembly. In fact, there was as people know, we had an announcement at the White House to make. It was going to be announced as soon as the first assembly was completed. We kept getting calls from the White House asking if it was soup yet. And I would call Soren and Gene. And there were a few glitches. The program crashed a few times, and I think about 3 hours before the White House announcement, it finally went together. So, by the year 2000, there were only 47 genome sequenced, but they covered, all 3 branches of life and slightly more completeness. By earlier this year, that jumped up to over 320 that were sequenced. And now there's literally, this was just an early count, thousands of genome projects going on. In a while, I'll tell you about just one side project we're doing on 160 genomes in 1 year. So after we finished human, and we did a few other species like mouse and Anopheles mosquito, rat, dog, we wanted to apply this on a broader scale. I had been fired from Celera by this time because even though we sequenced the genome and raised $1 billion, it was clearly not economically viable to continue. But I wanted to apply this back to the environment, and we tried a test experiment in the Sargasso Sea where we just took a barrel of seawater, filtered it, and shotgun sequenced everything on the filter, and we ended up discovering around 1.2 million new genes and literally tens of thousands of new species. And this kind of changed the view of the environment a little bit and led to the start of the Sorcerer II expedition, which the rationale for this was instead of sampling over and over again in the Sargasso Sea, we might find different organisms that were dominant in different areas. We didn't know the extent of diversity. And besides that, I always wanted to sail around the world on my own boat. So this was the ideal way to combine the two and prove that science is indeed pleasurable to do. The expedition took 2.5 years. We started in Halifax, went around mostly around the equator, as you can see. But all the data that's in the next set of papers and analysis, which took close to 2 years to do, is just from Halifax down through the Galapagos. This is our research vessel, the Sorcerer II. It's a 100-foot sloop. And that was, in fact, the best thing to come out of Celera besides the human genome. This is our scientific apparatus, simple millipore filtration, where we just pump a few hundred liters of seawater through a series of decreasing size filters, starting with 3 micron, then 0.8 micron. Most things that have been analyzed to date have been from the 0.1 micron filter. Everything that goes through that was collected on a tangential flow filter, which is primarily the viral fraction. It was a very simple protocol. After doing this, the filters went into the freezer. Whenever we reached a port with FedEx, we'd sent them back to the laboratory where they got processed. So we don't need 300 machines anymore because the current machines have about now close to 10 times the output of the machines we use for sequencing the human genome. And this technology is changing very rapidly, as you have heard and will be hearing further in this symposium. This facility does roughly 200 million letters of genetic code a day, but now with 454 technology and the latest version of that machine, each one of those machines can obviously roughly do the equivalent of this entire facility. The trouble is, as sequencing technology gets faster, the reads get shorter and shorter and less accurate. So it creates a lot of new problems mathematically for dealing with them. And an individual sequence read doesn't have the same value if it's only 10 base pairs, as some technology provides. 454 is now up to 250 base pairs. But when we're used to getting 900 base pairs of highly accurate data, it makes a big difference. But I think the technology is changing quite rapidly. So these are the actual sites from the initial analysis. Samples roughly every 200 miles. But those of you know the different environments. We have estuaries, including the Narragansett Bay here, open ocean, Gulf Stream, Panama Canal, and extensively around the Galapagos. So I figured I had to show this group some assembly statistics. So a little less than 8 million sequences, about 6 billion base pairs from 41 samples. And here's where it got quite stunning. About half the data had no overlap with any other sequence. And 85% of the assembled data is unique to each site. So this was really stunning to everybody and was not what any oceanographer or microbiologist predicted. So only about 3% of the data assembles over multiple sites, but that data is interesting on its own, and so we can compare the red here is hot, or the darker colors if you're colorblind, the blue and the lighter colors are cold, and just looking at the assembled data, there's a clear delineation. If you're using hybridization technology, whether it's an Affymetrix chip or others, this is all you can measure, the roughly 1 to 3% of the information. The rest is totally lost with hybridization technology. So sequencing is pretty critical to look deeper. But even with this, the 3% that can be looked at that level that assembles across sites, it's region specific. And so we can actually tell from a sample of seawater, for example, from the ballast water of the ship, where it originated. And the water and the microbes are unique to these different parts of the world. This is from the original paper on the Sargasso Sea. This is looking at new photoreceptors, and this is one of the many stunning discoveries. So the blue at the bottom there was the entire repertoire of photoreceptors, including our own visual pigments. There was only 2 bacterial rhodopsins that were known at the time. A group in Monterey found, working with TIGR, found a single photoreceptor in a back isolated from the ocean. And when they made PCR primers, that's the yellow section in the center, they found a lot of closely related photorhodopsins in that area. But when we just randomly select from the ocean from these filters, all these red ones are all the new photoreceptors that came out of the initial sample. And you can see there's very deep branching in these compared to others. The discovery of these has gone on in a linear fashion. Statistically, what it represents is probably every organism in the upper branches of the upper depths of the ocean probably have these photoreceptors. These are the 7 transmembrane receptor category, and it's light activated proton transport across the membrane. Not only were these not expected, but life basically was not expected in the Sargasso Sea. It was viewed as a desert because there's fundamentally no nutrients there. And now it's clear how all this life and all the diversity exist because they can get their energy straight from the sun. They don't require nutrients in the ocean for their survival. As we continue to collect these new tools in bioinformatics, such as multi-protein alignments, are becoming a real challenge. Just early on, it took 22 pages just to align these. When we had on the order of 2,300, I think we have close to 8,000 new photoreceptors now. But there's good reasons to do this. For example, with the photoreceptors, you can get down to a single amino acid residue that determines the wavelength of light that these receptors see. So looking at these, we can ask a lot of different questions, like are these differentially distributed in the environment? And it turns out they are, in what seems like a logical fashion. If you've ever been out in the Sargasso Sea, it's a deep indigo blue. All the photoreceptors on the organisms there see primarily blue light. If you get into coastal waters where there's a lot of chlorophyll, the photoreceptors see primarily green light. Fresh water, such as the Panama Canal, it's almost entirely a green light. And so what this implies is this from the single amino acid chains, this is the ultimate in Darwinian selection. If you have a different color photoreceptor, that very much affects your survivability in that environment. So the ones with green photoreceptors were dominated by the ones with blue receptors in the Sargasso Sea, for example. And this data gets even more interesting, as I'll show you in a while. The other surprise that's coming out of this data is the diversity of life greatly exceeds anybody's even imagination. So this is a bioinformatic tool developed by Doug Rush at the Institute, where you can take a sequenced genome and then literally throw all the data from the environmental sequencing against that. So each one of these little bars represents a 800 to 900 base pair sequence. So obviously, if there was an exact match, it would be up there at the 100% level. You see this band of 90 to 100% of highly similar sequences. So these are color-coded by site. Then we see this diverse band, roughly between 55% sequence identity and 90% sequence identity. And this was really stunning because, you know, when you get up to even 20 or 30% sequence divergence, we're talking about tens of millions of years at least. Because of these, a given bacterial gene is roughly 900 base pairs. So if you have a random sequence from the genome of 900 base pairs, it hits on the average of 1.4 genes. So a single sequence, you can actually discover 2 genes with it. But it also gives you neighbor analysis, because now you know what the neighboring gene is. So from this simple kind of plot, we can work out whether that gene has the same neighbors as it has in other genomes or in the one you're comparing it to. And it turns out the gene order is largely conserved over even this tremendous sequence diversity. So we have organisms now that are perhaps delineated hundreds of millions of years ago that still coexist in the same environment, have largely the same gene order with a lot of significant exceptions, but 50% sequence divergence. I don't think anybody would have predicted that. It actually gets interesting, and we had to develop a new tool because of all this sequence divergence. Even though there's a lot of sequence coverage for related organisms under a single 16S RNA type, they don't assemble because of this sequence divergence. So we created another tool we call extreme assembly, where we can take, by just loosening up the parameters using the Celera assembler that Soren helped to build, we can get assemblies that wouldn't normally go together. They're slightly looser clusterings, as you can see, around 98 to 100% here on the top. And this creates new now assemblies for us to compare sequences to, because there was only a few organisms that had been sequenced that there were any matches to at all. So this is one of those extreme assemblies. And here's some totally site-specific sequences. But again, it's not a single tight band. It's plus or minus 10% in sequence of variation. And then below that, we have this other widespread divergence. We see this for every and any organism, any sequence that's been compared. Sometimes these assemblies will have a 16S RNA in them, so we can link them back to a group such as Prochlorococcus, one of the SARS, etc. Most of them are unknown, but just from this pattern recognition, you can see how much diversity there is in sequence space around even similar parameters that we've seen before. And now we can start to do all kinds of new things. So with SAR11, it was supposed to be the only organism in the Sargasso Sea, basically. It's clearly not a single organism. It may be 10,000 different organisms, all with an identical 16S RNA, with sequence variation and genome content variation. So we can take this data and we can look at it in a number of fashions. For example, we can just take a slice at any point and then construct phylogenetic trees from that data. So you can see, just taking that information, we can see deep divergence on it. In fact, we can construct these so you can put any parameter you want there for looking at sequences that are Atlantic specific or found only in the Pacific. But the bottom is one of the most fascinating ones. This is looking at the photorhodopsin tuning, showing that it has switched back and forth at least 4 times in evolution from blue to green and back again. So here's the single amino acid residue that determines fitness for a specific environment, and it wasn't a one-way switch. It switched several times, probably as the environments have changed. So you can substitute in these plots anything you want to look at. The other thing we see when we compare to a reference genome, sometimes there's huge gaps, and they provide unique information on their own. In this case, it was a viral insertion that fundamentally wasn't matched in the environment. So because we're sequencing the DNA and RNA isolated from the bacterial fraction, any viruses that are there are by definition from infected bacteria. We can assemble a lot of the smaller phage, even using extreme assembly. But we can do other unique parameters with them, for example, looking at the ratio and the distribution of the phage versus their attacked organism. And we can work out even ratios and estimate in this case that about 3% of the cyanobacterial population was infected by this phage. But then when we look globally, we were again stunned by finding about a 20-fold variation in phage infectivity. This is totally new. It was not known, and it's thought that bacteriophage in the ocean are the primary determinants of turnover of microorganisms. And these are the same organisms that are responsible for most of carbon fixation in the ocean. So there's implications if you can understand phage infectivity and control it, you can dramatically change carbon fixation. The Sloan Foundation gave us a grant to do the air genome project. In fact, it shows you that we encourage people to go sailing or take up other sports because you get good ideas when you're doing this. So we were sailing from the Canary Islands to the Caribbean following Columbus's second route. And we were in the middle of the Atlantic Ocean, and we were engulfed in a sandstorm, 1,800 miles from Africa. The deck was actually covered with heavy sand. So our region, if sand could travel thousands of miles, that bacteria and viruses could travel globally. And that's where we got the idea to start sequencing the environment. And we use a very similar protocol. We have a sampler that captures things into a liquid phase that we can then take through the same filtration and sequence everything on the filter. So, and we can just apply the same Celera assembler to this kind of data. I can't give you too much in terms of specifics right now from this because we promised we would let some of the authorities know before we publish the data. But you'll be absolutely stunned what's in the air you breathe. Maybe you wouldn't be stunned what's in the air in New York, but we've also done sampling in Washington. We find everything from human DNA to pathogenic viruses to a variety of different environmental organisms depending which way the wind's blowing. If it's blowing off of farms, you see soil organisms. If it's blowing off the ocean, you see primarily ocean environmental organisms. So in a place like Providence with the Narragansett Bay here, you're going to obviously have a constant enrichment of the environment from both directions. So we basically live in a microbial and viral environment. There's 10 million viruses per milliliter of seawater. In the air, even during this lecture, particularly in a closed room like this, during this time, you're breathing in millions of bacteria and millions of viruses. We just published from TIGR the human gut genome project, where it looks like there's more microbial cells in our bodies than there are the 100 trillion human cells. So we're in a continuum of bacteria and viruses. Despite how many sterilizers you buy, you can't get away from it. Fortunately, very few of these ever become pathogenic, and most of them are essential for life. So out of all the gene discovery, questions you might all have, what's the novelty added by the data? Is it like the rhodopsin, we're just finding distant relatives of known families, or are we discovering anything new? What's the rate of discovery? Do we know most of biology? Do we know most of the gene pool? Are we early on? Just to show you impact of this data, just from Halifax to the Galapagos, the GOS data set, Global Ocean Sampling Data Set as we're calling it, is roughly twice the size of GenBank in terms of numbers of proteins. We've done an experiment that took over 1 million CPU hours, taking every bit of gene and protein data we could get out of GenBank, off of other websites, all the TIGR assembled EST data, assembled all this together with the GOS data to try and look at how many protein families there are thus far on Earth. And it comes around 40 to 50,000 major gene families, but any new sample from the environment adds new gene families in a totally linear fashion. And it doesn't matter whether we're looking at minor families or major ones, there's no hint of saturation except in certain phyla. Obviously, from the ocean, we're discovering primarily prokaryotes, whereas GenBank is heavily favored with eukaryotic data. But this Venn diagram has been the one that's, I think, surprised me more than anything else. The largest circle there is the GOS data. The small light blue circle is GenBank. There's roughly 115 major gene families in GenBank that we haven't hit with the ocean sampling. We have close to 4,000 new gene families with more than 50 members of them that have never been seen before with other sequencing. And the same is starting to happen with viral and eukaryotic sequences. This non-readable table is in the paper, and basically this is just more here as a reminder of people have been arguing they had phyla-specific genes and proteins. And basically, as we expand the data set, they all go away. So I think as we truly get a comprehensive view of life on this planet, I don't think there will be many, if any, phyla-specific genes and proteins. We use the same material over and over again. Now, obviously, we're up in the animal group, and there's been, since we sequenced human, there's been a lot of different mammals that have been done. And basically, you can see from this, the mammalian gene pool is pretty well saturated. Sequencing another mammalian genome won't add any new genes. It might add variants, some interesting structures, but we've pretty much saturated those, whereas the rest of the gene pool is linearly expanding. Gordon Moore Foundation has funded a lot of our work along with the Department of Energy, but they recently gave us $26 million to start a new database that's not just a database, and this is the so-called camera database that we're doing with UCSD. Well, I guess that's coming in a second. They also funded us to do 160 genomes, so we call it the Moore 160, of investigators collecting samples around the world and getting microbes in culture. I thought it was a good idea to sequence those to help put in context the global ocean sampling data. And in fact, I think it's put it in a very stunning context. So the rate limiting step is actually people sending us the DNA to sequence. But as we look at these environmental samples, it just shows with tens of thousands of different strains in the environment, taking any one thing into the lab and growing it doesn't give you a very good view of what's actually out there. In fact, those sequences, those genomes, don't recruit many of the sequences that we have from the environment. So here's the camera project. So it's unique in the sense that it's not just a data set. There's certainly not a sequence database. It's going to be a computational resource over the internet for this new environmental sequence data, where all of you will be able just to log on and do computations with the millions of new gene sequences going in there. But it's also going to have all the metadata, satellite imagery, the chemistry with the sites, the GPS coordinates, the even in many cases high definition video of different parameters of the site, and other findings as people add things on. So adding in 6 million genes doesn't take a lot of data storage. Adding in all the satellite imagery and other information certainly does, but we'll be able to make correlations that have never even been looked at before. And this is part of the new experiment for communication and science with the Lambda rail, and I'm sure this group is very familiar with it. I don't know if Brown is on the Lambda rail or not. You don't know. Oh. So what's happened with all this investment in fiber, we have all this dark fiber in the country, but what's changing faster than anything else in computing is data transmission. And so it turns out this fiber could be broken down into smaller wavelengths called lambdas, and a single lambda can transmit on the order of 10 gigabits of data a second. So many universities and facilities you can see here on the Lambda rail, and part of our funding, UCSD and the Venter Institute are now linked up with the Lambda rail that allows experiments have been done, for example, researchers in San Diego running an electron microscope in Japan and vice versa. It provides 10 times high definition television resolution in real time with no delay. So we actually have a table we're putting on a video wall that goes up where it looks like people are in the same room with you, and you can carry on real-time conversations, not these jerky images and delays that you see. And so I think it's going to start to change communication parameters, particularly in science where we're trying to use and move large pieces of information. So let me just talk about a few applications of all these genes and a project where we're now not just reading the genetic code, we're now entering into the era of being able to write it. And that's this new field of synthetic genomics. So with the GOS data, we have on the order of 10 million genes in the databases by early next year. Within a year from that, that's probably going to double or triple again. And one way to think about these is the design components of the future. If you think about the electronics industry in the early 50s, we had resistors and capacitors, the start of transistors, and look at all the different things that have been built out of these various components. With tens of millions of components, it's basically a limitation by imagination. We're trying to use this to understand basic cellular life. As probably many of you know, most genomes have a large percentage of genes of unknown function. Even with the smallest genome, Mycoplasma genitalium, over 150 genes are of unknown function. In our minimal predicted genome, 100 are of unknown function. And we're increasing the pool exponentially of unknown genes, so we need new ways to resolve these. This was the second genome in history. It has only 482 protein coding genes and 43 RNA genes. And we just ask simple questions. How many of these genes are essential? Can we get to a minimal operating system of a cell? And ultimately, could we construct it as in chemistry, where you synthesize the compound to prove the melting point is what predicted. The ultimate in having a sequence is, can you go back and reconstruct the genome to prove that you in fact had it right. We're taking a number of approaches, comparative genomics, we're doing gene knockouts, and then ultimately the synthetic genome. So this was actually the first comparative genomic paper by Koonin right after we published the first two genomes. And what he found is what others have done since, is there are only a little over 200 genes in common between these. So that wasn't particularly useful. We've gone on to compare 13 related mycoplasmas. And what was found is all of these have only about 173 genes in common. You can narrow it down a little bit by eliminating an intracellular parasite. It goes up to 220 genes in common. And then if we look at non-orthologous displacements, we can expand to a core of 310 genes. But based on a lot of other observations, this is probably not sufficient to sustain life in an independent cellular fashion. We've also done knockouts. So we've developed, Clyde Hutchison at the Institute developed the technique of whole genome transposon mutagenesis, where you put in transposons, You see what survives. Then with a PCR primer, you sequence right off the transposon, and you find out down to the base pair where the transposon inserted. So all these triangles are where they inserted in the genome. Genes that took transposons we deemed non-essential. The ones that the cell couldn't survive with transposons, the ones without them here, we deemed essential genes. But what's not known is, can you do this on all these genes simultaneously that takes transposons, or there are genes that compensate? And we know there's compensatory ones as well as environmental definitions. A simple example is the cell has two sugar transporters, one for fructose and one for glucose. If you have both sugars in the environment, you can knock out the glucose transporter and you would label it as a non-essential gene. If you only have glucose in the environment, you knock out that transporter, you would call it an essential gene. So everything has a conditional definition, and it turned out to be one of our most naive assumptions that we could ever define a genetic code for life. There can't be an operating system without defining the corresponding environment. But if we look at all the pathways that we can knock out individually, it looks like it'd probably be a pretty sick cell. So this was not very satisfactory as an approach either. And so we decided the only way to answer these questions was to actually build the cell from scratch. We're doing this two ways. We're trying to isolate all the different proteins, see if we can assemble them together with a little lipid and chromosome and maybe a lightning bolt. And the shorter term approach is we're making a synthetic chromosome. We plan to reprogram a cell with it. When we started this, we found it's almost impossible to accurately synthesize DNA. Even oligos, it's a degenerate procedure, and so assembling lots of oligos certainly doesn't make it more accurate. You can do that with sequences, you can't do that with oligos. So Ham Smith and Clyde Hutchison developed this approach where we could take a sequence out of GenBank and within 2 weeks have an assembled sequence, in this case for Phi-X174, that we injected into E. coli, and the E. coli started manufacturing Phi-X174 phage, which then started killing the bacteria, and that's what you see, these clear plaques. So this is now a case where the software builds its own hardware. So it's like you write a computer program and it goes out and builds the computer around it. That's what biology does. So it's pretty stunning, and it's a question of what the scale is as we switch from reading to writing the genetic code. Right now, and this is from a Senate testimony I gave a year ago, any sequenced viral genome today can be synthesized. Hopefully designer viruses are a long way away. The first synthetic bacteria will exist within a year, and probably synthetic eukaryotes within 5 to 10 years. Imagine what this does to agriculture if you can design the characteristics of a plant you want and make it, not just take what's out there. The third synthetic virus after polio, then Phi-X, was the recent synthesis of the 1918 flu virus, and this is the first true Jurassic Park scenario where the RNA was isolated from the lungs of a woman that was buried in the permafrost in 1918. The RNA was sequenced, and then using techniques similar to what I just showed you, the virus reconstructed at the CDC and shown to be as infective as predicted. But this is very key because from the sequence, it was not obvious what portions of the sequence caused the extreme virulence of this virus. So these are important experiments because they're now helping with viral screening and vaccine development. We're trying to build the artificial chromosome in a cassette-based fashion of 5 to 10 genes so we can do shuffling, check the importance of genome order, gene order, etc. And the initial stages, we're trying to just replace an existing chromosome with a synthetic one using a number of different approaches. And this is looking very promising to actually transform the entire phenotype of a cell by changing the operating system, changing its genome. So it's like changing your genome and putting a different species chromosome in, and all of a sudden you take on the characteristics of that species. We're trying to do this on large scale, and we're trying to do this by setting up automated homologous recombination to build things from the smaller fragments. And we're doing this by trying to isolate the repair system from Deinococcus radiodurans. So this is the organism that can take 3 million rads of radiation and keep on going. This is the chromosome in the top panel after 1.75 megarads. It really gets blown apart, a couple hundred double-stranded breaks. And the bottom one is 24 hours later. It reconstructs the genome. So this is the biological equivalent of the Celera assembler. It works robustly, rebuilding it the same every time. So if we can isolate this and get this to work, we'll have a new field of combinatorial genomics where we think maybe we can have a robot that can make 1 million genomes a day. And then you just screen those for different parameters. You can screen for viability by, if you rearrange gene order, does it still live? And then you can start to screen for other more interesting parameters. And some of these are very important in terms of applications to the environment. We're adding 3.5 billion tons of CO2 to the atmosphere. The world population, even with HIV, is still growing, but it's going to be more like 9 billion instead of 12 billion people. There's a challenge in feeding those. The energy demand is increasing substantially, particularly as the developing world wants to come up and use the same technology that you all have access to. You can see the oil use is going up. It's thought that though this is going to saturate and then start to decrease in the next 15 to 75 years, depending on predictions. So we need alternatives for fossil fuels for lots of reasons. And I got the first idea from this when we sequenced the first archaea. This is the first autotroph that was characterized. It doesn't use any organic compounds. It just takes everything from CO2 to methane, and that's how it generates its ATP. At our body temperatures, it's frozen. Its temperature optimum is 85 degrees centigrade, and this is all driven by hydrogen. So we're designing synthetic cells to make fuels, ideally capturing CO2 as the source of carbon. DuPont has an experiment, a pilot plant in Tennessee to make propane diol from sugar. They're literally making hundreds of tons of this by an engineered bacteria they spent the last 10 years engineering. Oil was the source of the carbon. That can no longer be the source of carbon, so companies that were built to use that source are now beginning to switch. We have a lot of energy that hits the surface in terms of terawatts of energy from the sun. We're trying to find ways to capture that. We're modifying photosynthesis to take the electrons right into hydrogen production with oxygen insensitive hydrogenases. And then there's this whole notion of cellulose into fuel. Right now, a lot of the concentration is on ethanol, and we have a team making a cell that not only can have cellular cellulases on the surface to break down the cellulose, but then in the same bacteria ferment that into alcohols. There hasn't been this notion of energy crops other than people burning firewood for a long time. So we can expect as we go into this era of energy crops a lot of gains, particularly with the progress in molecular biology. So we're working on trying to capture the cellulosic sugars out of different feedstocks to convert those into a variety of substances. But we are primarily working on the cell that we think can go from sugar right into octane or some other highly burnable fuel. So it could be a bacterial cylinder you carry in your car and feed it candy bars. So future uses of this, obviously understanding cellular life. Within a decade, you're going to see a dramatic change in the petrochemical industry, hopefully new major sources of energy, bioremediation, and then through imagine cassettes of different parts of immunoglobulins doing rapid recombination to produce large amounts of vaccines. We started ethical review of the synthetic project back in the mid-1990s. The results of this were published in Science in 1999, saying that it was acceptable for us to proceed for the reasons that we were going forward with. Yesterday, we just finished a year-long process funded by the Sloan Foundation with MIT, trying to come up with guidelines for this field. I'll end with, this is our Discovered Genomics program, so we're trying to capture the hearts and minds early. We go to middle schools and have kids do exciting experiments, including solving crime scenes. They're doing experiments with DNA fingerprinting, isolation, other things that I was a full professor before those were viable techniques, and the response has been pretty overwhelming as hopefully we enter this new era. Thank you very much.
We are supposed to have a sweat box session. In the interest of time, let's have it here. So let's have questions now.
Does that mean you turn up the thermometer?
It will be a little bit less tough questions. Yes?
Do you think it's more likely that we use this technology to improve ourselves or to change the environment around us that makes it better for ourselves?
It's a good question. I think, do we need to repeat the questions? Can people hear them in the other rooms? Which? No, okay. So the question, are we most likely to use this technology to improve ourselves or the environment around us? I think both, but the first will be improving the environment around us. You've heard some people argue that the only long-term survival for the human race is to go and mess up another planet somewhere else. Others have argued the only survivability is to change our genetic code and change our environment together so that we can survive. I think both are inevitable. It's a question of the time course. Yes?
Have you been in discussion with the third world countries in terms of how these technologies can improve their livelihood, not even the livelihood. Like you mentioned China and India as two countries which are actually taking up all this energy, and it is going to take energy. What is the impact of this in those countries?
So the question is, what's the impact of this technology on the developing countries? And it's actually where we think there will be the most impact. So if we can take cellulosic sugars from palm trees, from jatropha, from bamboo, from a variety of sources that are out there, it will change from the oil-producing countries to the agricultural-producing countries. So basically, the countries that are now the developing world will be the sources of fuel and have equal change, possibly an economic impact that oil's provided the oil-rich countries. So there'll be, imagine the economic and social shift, political shift, if that happens. Yes?
So you used rhodopsin to illustrate diversity. Is that because you never met a G protein you didn't like from your early career, or did it emerge? And if it emerged, what does it tell us about regulation and processes? And what didn't you tell us about that also reflects diversity? For instance, receptor tyrosine kinase.
Yeah. It's not clear what the single question is there to repeat, but it started with rhodopsins. It was simply a stunning observation from looking at the proteins that popped out of this environment. Obviously, we ask questions where there's 7 transmembrane molecules there, but we are trying to understand why and how there could be such huge diversity of life in an area that biologists had claimed was a desert, and there was and should be no life. So we started looking for things for energy sources, and we discovered those. And so the challenge, in those of you who start to use the GOS database when it comes online early next year, there's probably 1 million similar discoveries remaining to be made there. With a small team, we can only ask so many questions. So we tried to pick things either from our prior experience in molecules we knew how to recognize, to ones that could answer very specific questions. So there are some gene families now in this data set where there's 30 or 40,000 members. So if you're developing new multiple protein alignment tools, they're going to have to be very robust, and we have to think of new ways to visualize how do you print out a 30,000 protein alignment. So, we're just making discoveries that were obvious looking at the data of things that popped out that were unexpected, given the repertoire of the people looking at the data. A different group of people would have found totally different discoveries, and probably will. Yes?
Is there a fundamental premise that organisms you can design from scratch will be more useful than ones you can modify?
Absolutely. So the question is there a fundamental premise that if we can design something from scratch, it'll be more useful than what you can find and modify? Absolutely. People have been looking for energy-producing bacteria probably for half a century at least, if not more. Hydrogen producers, for example, and it's been shown that you can change metabolism 1 million-fold or more through genetic engineering.
From scratch, you mean you're going to take existing genes and put them together? Or you're going to design absolutely original genes?
Well, if we have a repertoire of tens of millions of genes, I think coming up with totally new ones is probably not that critical at this stage. As I said, we're limited more by imagination than by the gene repertoire we have to work with, but other people in the synthetic biology field have come up with, relatively new genes in the sense of changing codon usages. I mean, in theory, you could design a cell that uses a totally different set of codons.
But getting even 500 new genes that haven't evolved together to work together, maybe not just because I'm not as optimistic as you, I view this as almost impossible.
Well, you'll be one of the happy, stunned people in the near future. We like those kind of people.
Even if you could predict how proteins will function, which you can't. Predicting how they're going to interact with each other on a 500 by 500 matrix, just one terrible interaction will kill that cell.
Well, obviously,
even for you may have picked a mountain too high.
Well, those are my favorite kind of challenges. So it's, but all we have to do is look at the diversity of life. In fact, we've come to think of the planet in a gene-centric way, not a genome-centric way. And I think it's a matter of just coming up with different combinations. I don't think the regulation is so tough. That the mycoplasmas have no regulation. They're either on or they're off as a set.
You're trying to do, granted you have the human mind to guide you, you're trying to do in 1 year, 5 years, 10 years, what took evolution millions of years, trial and error.
Well, I don't have as much time. So we have to do something better. But that's why, we're at the stage, I mean, ideally you'd want to do all this based on first principles. We don't know too many first principles of biology that apply across biology. I think only one that I can think of is what we derive from sequencing genomes, that each species genome has a unique mathematical equation. Otherwise, what we do with genome assembly wouldn't be possible. And that's why people thought it couldn't possibly work when we go to the environment, that things couldn't assemble, not for the reasons they're difficult to assemble, but just because the algorithms couldn't recognize that degree of uniqueness. But that's why the empirical methods are necessary. If we make 1 million genomes a day and you can select for viability, I only have to be right one time out of 1 million.
Yeah. But
Right?
if the number is 10 to the 29th, then you're never going to be right.
Stay tuned to this channel.
Okay.
John, are you worrying about protein misfolding? That's what you're talking about? John?
Sorry?
Are you worried about protein misfolding? That's what
Not just protein misfolding, even if they folded perfectly, which is probably not going to happen. the odds of just picking proteins and then interacting, playing nicely with each other. I think that's one of the great overlooked qualities in evolution, is that proteins are selected not just for what they do, but for what they don't do. And mostly it's what they don't do, which is interacting negatively with other guys. So that's really, I think, the biggest problem.
In fact, that's why we're starting with a minimal genome. What DuPont had to do over 10 years was shut off metabolic pathways, not add them in. So if we can start with a minimal matrix and add function to it, we can do real-time evolution.
Yeah, I think it's incredibly cool. I'm not arguing at all with the premise. It's real sketch.
Can you comment on this ethical issue of these opportunities here are tremendous, but the dawn of genomic terrorism could bring that on? Can you just comment on that might impact what might
Well, I feel in my career I've been subject to genome terrorism from scientific reviewers, but obviously that's the number one concern that people have, because, you know, in what came out at the meeting yesterday, this is a U.S.-centric point of view because we live in a terror society. The European view was very different. They were much more concerned about environmental release and other things. The U.S., the primary concern seems to be bioterrorism. You know, we don't have an evidence-based system in our society. We have a fear-based system, and so quite often, I think fears can result in self-fulfilling prophecies by stirring up and stimulating people to do things that might never have been done otherwise. I think we do have to be concerned with it. But as we saw with SARS and we saw with other recent emerging diseases, we have a problem that we have to solve with the expanding population. We still can't treat AIDS very effectively. We can delay it, but we can't reverse it. Of needing new antivirals, new antibiotics, just to deal with the threat of existing on this planet. And there's really no difference if it's naturally occurring or human-made. We have a problem of a society we have to solve. I don't think it's a reason to stop the progression of science. In the back.
Speaking of diversity, people are even able now to make organisms with more than the normal 20 genetically encoded amino acids, like Peter Schultz has done.
Right. That's what I was referring to earlier with Schultz's
Is
work.
there a limit to how much diversity even you are willing to accept, or you think that's pretty cool too?
So the question is, are there limits of diversity? You know, and the reference was made to Peter Schultz developing a new codon for a new amino acid. I think, you know, while we're currently limited by our imagination, at some stage we'll be limited by reality. Ham Smith and I and Clyde Hutchison were discussing yesterday, could we design a D amino acid based organism that'd be a mirror image of another one? So you can have all kinds of fantasy questions. You know, it's not clear. Some people have advocated we should only use artificial amino acids in synthetic biology, so something can't exist in the environment. But I think there's enough other ways to prevent things from escaping. But I don't know. Right? It's a trial and error empirical stage right now. Steve.
For years, you've been talking about how close races are, humans to chimps, and a few weeks ago we had a publication in Nature which sought to differ with that perspective. You want to comment on that?
Well, in fact, I didn't even talk about our new human genome work, so we've gone on to do a whole new version of the human genome, the first actual diploid genome. And we agree very much with the study that was recently published that only looked at even a small portion of the diversity that's out there. When it was first talked about how close humans and chimps were, it's from the areas of the genome that align nicely. And that was less than you know, a percentage difference in general. But when you look at indels, humans and chimps differ by close to 6%. And it turns out when you start to look at true human variation, humans differ a lot more than 1 out of 1,000 base pairs. You know, the SNP-centric view of biology is just so distorted thinking, it's missed probably the other 90% of the picture. and that will become clear with time.
So, what's your view regarding personalized medicine and the difference between humans?
The question was, what's my view on personalized medicine? So, Victor McCusick makes the distinction, he likes to call it individualized medicine versus personalized. I don't think drugs will ever be made for each one of us, but it's very clear there's genetic variations that cause, susceptibility to effectiveness, lack of effectiveness. Most drugs only work on a third of the population. There's another third that they don't do any harm to, and another third they do harm to. You'd ideally like to know which class you're in when you take a drug. And that's why I agree with Victor. It's much more individualization of things in terms of knowing your genetic repertoire and knowing what works for you and what doesn't versus something being designed for you. This notion of one unique drug for each person, I think, is just silliness.
It goes back also to animal models of human diseases. So we just have an example of Pfizer losing $2 billion or $3 billion in one day. And the issue is, well, we sequence this genome, the dog genome, the mouse genome, but there might be great variation between mice and dogs, etc., etc. So when we test human diseases in model animals, we have the same problem.
In fact, the loss of the Pfizer drug is an important one. I had high hopes for that one, a drug that increased HDL levels had promise, at least in terms of reversing heart disease. But it shows why the pharmaceutical industry is such a crapshoot. You're better off going to casinos. Yes.
To what extent is your method of work empirical, that is checking anything that is checkable, versus theoretical, that is deciding exactly what and how you want to do your process?
Well, it's obviously a combination of both. In the case of the environmental sampling, it started with the theory that resulted in widespread empirical testing. And it was the empiricism that gave us the new findings. We certainly even the notion of being able to assemble and find genetic diversity through just sequencing DNA was a concept we couldn't get initial funding for until we went out and did it. So, but I think that's one of the values of computational approaches. They narrow down the set of questions that you have to ask. So it's not starting with just absolute empiricism and hoping you bump into something in a lucky fashion. We can perhaps get much more sophisticated about the questions we're trying to answer and ask them in a much more intelligent way by starting with computational approaches. I think we're stuck with empiricism for a very long time. Yes?
So do you think there are direct applications of environmental sampling, taking all these genes from various sources, to synthetic genomics?
So the question, do I think there's direct applications from the environmental sampling to synthetic genomics? And the answer is absolutely yes. It increases our parts department exponentially. So in fact, dealing with the question about protein diversity, we have 30,000 different ABC transporters. If one doesn't work, it's not that hard to try all 30,000 to see which one does. That diversity, that parts department, I think, allows us to empirically go after finding the right combinations. Yes, ma'am.
That goes back to what the gentleman in red said. It's fine to just swap out the 30,000 and multiply the number of genes. When you take the power of the number of genes and the number of possibilities, it gets astronomical.
Okay. Well, the comment was, these are astronomically difficult things to do.
It's not that it's difficult. It's actually very simple, but it's the total number of possibilities may exceed. Maybe if you start with your minimal genome and then just play around with it. It's just that the numbers get big.
The numbers do get big quickly, but I think it gets I think one of the true principles of life you know, in a simplistic view, is it's so robust, life happens. I don't think it's so complicated. I think things will want to work in a robust fashion and adapt quickly to do that.
Well, then there would be other life forms on Earth that weren't DNA, RNA based, if that was really true. If it was so easy.
No, it doesn't eliminate DNA or RNA. It actually,
No, but I mean, if life was easy.
Well,
Then why would it have to be DNA or RNA? It doesn't, right? It could be other self-replicating molecules.
well, I mean, given the repertoire of chemicals that we have, I think we'll get the same answer over and over again. But I don't think we have any idea how diverse life is and how many. You know, we have such a simplistic view of the repertoire.
All your data, your wonderful data, indicate is that there's still one common form of life. There's no evidence that any organism arose separately from the primordial cell.
Well, you can't have it both ways. It can't be simple and impossible.
Which of us is having it both ways?
I always try to.
I was wondering what you think should be done to combat efforts to produce synthetic or designer pathogens.
Well, why do you want to do that?
I certainly don't, but
I think we should lock you up if that's your intent. You know? I think the motivation to develop, you know, new pathogens, you know, hopefully is pretty limited. If you want anthrax, go out to any farm and get all the anthrax you want. You know, what do you need a new one for? So I think, I mean, some people in this new emerging field have urged biological hacking, and they hope that people will design new life forms in their garages and, you know, be the first one on your block to build a virus or something. But I don't think that's going to be very effective. And when you look at the diversity of things, the few number of organisms that became human pathogens, not likely to be very successful. I have a high regard for humanity in general, and I think history has shown there's not too many people motivated to do those kind of exercises. Yeah.
How diverse do you think the genome repertoire and the protein repertoire are? If you go sail around the world again and sample in the same spots, will this be reproducible, or you get a whole new set, an equal size set? How many times do you have to go around before it starts?
Yeah. No, it's a good question. So the question is how much diversity is there, and if we do repeated sampling, will we find the same thing? And this is a squabble I had with oceanographers who like to go out to a fixed set of GPS coordinates and do repeated sampling there. As a sailor, I'm very aware that water moves. Right? So it's like, look at this. I discovered this upstream versus downstream. You know, you wait at the bottom and it'll come to you. I mean, but reproducibility is important, and what was shown in the Sargasso Sea by sampling winter and summer, there was certainly a variation of organisms. There's different upwellings. But if the question is what does it take to saturate and understand the complete environment, I'd say we've moved up from maybe 0.1% of knowledge of life on the planet to maybe a couple tenths of a percent. I think we have so far to go that it will look like an infinite sink the first few times we do those experiments.
How about soil? Have you thought about doing the same?
Yeah, no, we are doing soil. Soil is a little bit more difficult in the sense that it requires an amplification step, taking a small soil sample, but we do have soil samples from a lot of these same environments, from sea bottoms, et cetera, that you know, the nice thing about random sampling and getting diversity is if the techniques don't require amplification, you get a cleaner answer. As soon as you have to do an amplification step, you lose the value of really understanding the stoichiometry at this stage. But we are doing soil, and people have argued for decades that microbial diversity in soil was maybe 100 or more times that in the ocean. I think our data sort of makes them more on par with each other. But, you know, we're stunned how much diversity there is in the air. So I think we have a very long way to go. I think any student today, it's the opposite of when I was a graduate student. I'm saying now any student can take a cup of seawater, you know, a cup of earth, and make more discoveries than previous generations have together. And the more people that do that, I think the more exciting it's going to be. Yes?
Sort of natural synthetic genome question. The real organisms we see on the planet are plants and animals that we can see visually, and those are synthetic genomes that came about from the mitochondrial and chloroplast and the symbiotic events. Mm-hmm. If there's so much diversity in the oceans and in the soils and so on, why is it that for 4 billion years all they have done is stayed as bacteria? Why have they not synthesized new genomes and reinvented endosymbiotic and eukaryotic cells?
So, I mean, it's an important but slightly off question. The question is, if we have so many billions or trillions of organisms, why do they stay as those, and why isn't it, just billions of bodies floating out in the ocean? Evolution, all you have to do is get one out of those to change and go to the next stage. It doesn't mean you eliminate the entire previous population. In fact, that's what's stunning about our data that's really confusing people, the coexistence of all these diverse, sequentially diverse organisms with basically the same gene content and gene order in it. Why wasn't there selection? Why didn't most of them get eliminated? Why isn't what people thought? There's this huge clonality. You go out there and you find SAR11, or you find Prochlorococcus. I mean, a few years ago when the Prochlorococcus genome was published in Nature, the leading line is, this is the most abundant species on the planet. We have not had an exact match to that genome yet with all our sampling. So, it's a more complex question. What we don't see in the comment over here is we're trying to condense millions and billions of years of evolution to a short time period. I think that time is the most complex thing to try and imagine or measure. We see all these things are at the same stage of evolution. We don't know what went through mutation and went to some other stage. I mean, I think we're seeing that with the Neanderthal genome data. We like to think those Neanderthals really had bizarre genomes and, big clunky heads and were stupid. They look like, our relatives, or maybe my relatives. Yes.
So if we're successful in synthesizing these new organisms to solve our huge problems, like extracting CO2 from the environment or producing energy sources, what's your vision of what that's going to look like in the future? Is it going to be some next generation of a catalytic converter that goes in your car and pulls out all the CO2? Is it going to be huge glass vats in the middle of Death Valley producing ethanol? I mean, what's your vision of where that's going to be?
It could be in your colon.
Well, some people have proposed huge glass vats in the desert to do this, to basically using sunlight to capture CO2, convert it into lipids, and then burn that as a renewable fuel. I think one of the challenges is if we don't use genetic engineering to increase the yield of various crops, there's going to be this huge dichotomy between what we use for biological energy in terms of eating versus what we use for fuel energy. I think it's very likely within, certainly 50 years that if this field is at all successful, burning oil and coal will be outlawed. And then it's a question of how much CO2 we need to capture back from the atmosphere versus getting that carbon in a renewable fashion from various crops. There won't be any one thing that solves the equation. It's going to be a combination of a lot of different diverse biology, solar energy, wind energy. My view and the view of many others, it's not an option to get there. If biology is only a 20% contributor to the solution, that's a pretty huge contribution. I don't think it has to solve everything. I don't know if that's what you were asking. Yes.
In terms of the prospects for making vaccines that you talked about, just drugs in general, do you see that as something that's going to be financially effective? Because even today in the developing nations with HIV and AIDS, there are some drugs that would be helpful, but finances get in the way in terms of distributing them. So how do you see that fitting in with the finances?
And that's really more of an economic argument. I think we have a false infrastructure in terms of pharmaceutical development and financing. That was the basis behind what Gates Foundation is funding in terms of this new anti-malarial drug, getting early genetic and synthetic biology to get a bacterial cell to produce the drug in abundance. It's not quite clear how regulatory issues are going to deal with that, who's going to actually produce it. But there's lots of other models other than the gross profit model for pharmaceutical production. I mean, so you can't assume that everything has to fit into that model in the future.
I think we have to take one last question.
So thinking about the applicability of some of these to medicine, and you've mentioned some of it relating to infectious diseases. So, for example, instead of sampling a bucket from the ocean, you can sample a patient who has a certain clinical condition and you don't know what he has, which is probably the case in more than 50% of infectious diseases. What do you think? Do you think this is applicable to this, and are you interested in working on it, or are you working?
Yeah. So the question is, what about applying these times environmental genomics directly to patients? And the answer is absolutely yes. There's huge applications. There's indication just looking at the oral flora that you can tell where somebody came from in the world, and there's implications for health with that. Looking at what's in the GI tract, other cavities, systems can predict things. Obviously, if somebody comes in with septicemia and you could get an immediate readout of the bacterial repertoire that's in the bloodstream, the challenge is it might be a lot more diverse than we think of single organisms and single strains, but at least gives data to try and make real-time decisions on, instead of waiting for a blood culture with the patient likely dying while you're waiting for the lab results. So there's totally going to be real-time things. With H. pylori, you can actually, which was I think the 4th genome we did, you can track populations of people totally by the strains of H. pylori they have in their guts, by the way it's passed on. It's more of a fingerprint identifier in many aspects than your own DNA is, but it also can be diagnostic and predictive. So I think we're at the earliest stages of this and the applications of it. And that's what these various technology companies are trying to scale up to do. How do you get that answer quickly enough? And some are trying hybridization, some are trying sequencing. Neither is adequate at this stage to really do it robustly, but the technology is changing rapidly.
So let's thank the speaker. So we'll take a short break, let's say 10 minutes, and then we